Today, we're moving into this process known as mitosis. We're going to produce some daughter cells. Now, the term mitosis is thrown around haphazardly and willy-nilly. You have probably, at this point, having taken basic science and public education, have heard of mitosis. How many of you guys have heard of mitosis? Everybody! Most of you heard of it wrong. It's unfortunate. There's a big misunderstanding about what mitosis is. Now, you have to understand, life moves in cycles. Was it Circle of Life, right? Lion King? And it's unfortunate. You guys remember The Lion King? Anybody not seen The Lion King? Because spoilers. So in the beginning of the movie, you have this baby lion born. This baby lion's named Simba, right? So they lift Simba up on this rock, and they say, "Behold, Simba, these are all of the things you're going to eat in your life." And all the things look at him and say, "Yes, I will be eaten by you." And over time, the mommy lion and the dad lion made that baby lion. And then Simba grows up, goes through, becomes an adult, finds a mate, mates with her, produces a new baby. They hold up at the end. It's a cycle. Frogs live in cycles. That's more like a spiral, I guess. But the adult frog lays eggs. The eggs grow into tadpoles. The tadpoles become froglets. The froglets become an adult frog. Cycles. The adult frog produces eggs and back again. There's one organism that quite literally lives in a cycle. It's a type of jellyfish that is functionally immortal. It goes from being an adult jellyfish, to being, so an adult jellyfish, you guys know what a jellyfish is? They look like an umbrella with tentacles. We're good on that? All right. The adult jellyfish is called the Medusa form. The immature jellyfish is called the... What's the immature jellyfish called? It's not the nymph. It's the... Well, shoot, it's slipping my mind. The larval form, whatever that may be. They look almost like a polyp. It's called a polyp. It looks like, do you guys know what bugles are? Like the food? Anybody eaten bugles before? What do you do with bugles? Witch fingers. Yeah. Witch fingers. You can also stack bugles on each other. That's what these juvenile forms of the jellyfish looks like. Well, they're these juvenile forms, and then they burst off. They sort of let go of each other, and they go floating free as medusas, and they get bigger and bigger and bigger. And then the adult actually starts to Benjamin Button itself and goes and becomes smaller and smaller and smaller and goes back to that juvenile form, and then becomes an adult again, and then back to a juvenile form, and just yo-yos. It lives literally forever by growing old and then growing young, and then growing old and growing young. And you have to admit that'd be kind of cool. Life moves in cycles. Now, if you recall, Rudolf Virchow put out this tenet of cell theory. All cells come from other cells, omnicellulae, acellulae. So today we're discussing how we get cells from other cells. How this actually happens. For 4 billion years, billion with a B, it's a long, long time, since almost the formation of the Earth, you've had cells and protocells making copies of themselves. Effectively, one organism looks like the organism that gave birth to it, that created it, with just some slight differences. You pretty much look like your parents, don't you? And I know, some of you guys are like, no, not at all. You do. You've generally, I might be making some assumptions here, but you've all got one head. Yeah. 2 arms, 2 legs, a digestive system. You breathe oxygen. You're not terribly different from your parents. They're not terribly different from their parents. They're not terribly different from their parents. But over time, those differences build up. If you look back several thousand years, you're going to look different than your ancient ancestors. They're going to look different from theirs. So over time, these differences build up and make things that are current look different from things that are ancient. So these cells have been dividing for billions of years. It's a highly regulated process. And you have to respond to the environment and be precisely timed. You want to reproduce at just the right time. Now for you guys, if you've ever thought about reproducing, what would be the right conditions to reproduce for you? I'm not saying the right conditions to mate. I don't want to know. I'm saying the right conditions to have an offspring. In your mind. When would be the best time? I know, it's not now. When is? I'm not trying to... Okay. Think about it this way. Don't think about yourself. Think about, I don't know, your best friend. When is the right time for them to have a kid? Never. Why? What? It would not work out. Why would it not work out? Give me a reason. He's crazy. He's unstable. There are elements about him that would make it dangerous. If the environment for the offspring is dangerous, you don't want to have kids. You don't want to have kids in a war-torn country, right? Why? Because you're trying to take care of yourself. If these cells are surrounded by antagonistic cells, cells that want to kill them, you don't want to reproduce. All of your energy needs to go into keeping yourself alive. What about other people? Do you guys have friends? When would you suggest that they have, or why would you stop somebody from having kids? How about that? Your friend is crazy. You don't want him to have kids. Why else might you stop someone? You might say, you know, now's not the right time. This is stupid. Don't do it. They haven't gotten their life situated. They haven't lived their whole life. They haven't grown. They haven't... Pardon? No career. Oh, now you've got two different things. So we'll start with, they haven't grown enough. They haven't matured enough. They're crazy. Cells have to reach a certain stage of maturity. And the next part, they haven't gotten very far in their career. Now, what's the point of that? With their career, what are they getting from their career that's important? Money. Resources. If the cell doesn't have adequate resources around it, it's not going to reproduce. Does that make sense? The same things that go into our minds as we think about, are we going to reproduce, are we not, the cell has to respond to as well. Now it's going to respond to all these things in this environment. Are these things present, are they not present? How is the cell going to figure out what's going on in its environment? What part of the cell is it going to use to figure out what's going on the outside? Yeah, the cell membrane. You got all those cool proteins in there that are acting as receptors. Now, inside of the cell, you've got the nucleus. And inside the nucleus, you have these chromosomes. Chromosomes are just super highly coiled up DNA. Remember that part? I just want to make you guys remember this. Because when I'm talking about chromosomes, I'm talking about DNA. And DNA is a nucleic acid that helps to produce proteins. As cells begin to divide, as they get the right conditions to divide, the nucleic acid, the DNA, moves from being chromatin, which is very highly decondensed, it's sort of all over the place, to being chromosomes. They're very, very tightly coiled, and you can see them. For most of the cell's life, they're these big open weave structures. That lets, I'm not going to get into it too much, but it lets proteins bind to them that can start creating other proteins. When they become really tight, suddenly those proteins can't bind anymore, so you don't get more proteins produced. Every species has a specific number of proteins. I'm not saying it's a unique number to that species, but what I'm saying is each species has a very specific number. Frogs have a total of 22 chromosomes, 11 of which match each other. They sort of are almost copies. They're very similar. Flies have a total of 8, with 4 of them that are very similar to each other. Humans have 46 chromosomes, 23 of which are very similar to each other. So here's our chromosomes. We've got 1 through 22, what are termed autosomes, and then the last set that's called your sex chromosomes. Typically, they're going to be lined up by size. This is called a karyotype. We're going to get into this in a minute. But you'll see that there's very long ones that are 1, very short ones down here at 22. Each of these is made up of DNA. So the longer ones have more DNA. The shorter ones have less. Most species, you and I, butterflies, things like that, are going to have 2 chromosomes that are going to determine our gender. For humans, for us, males are going to be considered X. They have an X chromosome and a Y chromosome. Females are going to be considered XX. So what does this tell you about the relative amounts of DNA that females have compared to males? Females have a lot more than males. Males have this little dinky little thing here. This is actually a lot of leftover genetic crap. Any chromosome that's not a sex chromosome is called an autosome. So everything but the sex chromosomes are autosomes. When your cell contains 2 complete sets of chromosomes, it's called diploid. Diploid, D-I. Like adenosine diphosphate. What does di mean? 2. So here we have a diploid set, diploid. 2 of the red, 2 of the blue, 2 of the yellow. They sort of match up. You are a diploid organism. Most mammals are diploid. In fact, all mammals are diploid. We're going to note diploid by writing 2n, where n equals the number of unique chromosomes. So how many unique chromosomes do we have? Absolutely unique. 1, 2, 3. So diploid is 2n. N equals 3. So total number of chromosomes is 6. 2 times 3. Oh, sorry. Let me walk you through that again. A diploid organism, so diploid, that means that it has a certain number of unique chromosomes. In this case, you have 3 unique chromosomes, red, blue, yellow. N equals 3. So 2n, just like in algebra, 2 times 3 equals 6. So most of the cells in your body, the vast majority of cells in your body are diploid. However, you do have a few cells that are haploid. Haploid means half. So instead of being 2n, they're going to be labeled as n. These are called your gametes, your sex cells. Females produce eggs in humans, males produce sperm. They have half the number of chromosomes. They are haploid. And that makes sense, because if you were to take, math prereq, right? A diploid organism and add it to a diploid organism, so 2n plus 2n equals what? 4, keep n, yes, 4n, because it's algebra. Don't forget that n, it's kind of important. And then, so if you have a mom that's 2n and a dad that's 2n, they mate, they produce 4n. These kids mate. 4n plus 4n is what? 8n. You're doubling the number of chromosomes in each generation. So instead what you have to do is you have to take that 2n and split it in half. So you have n for your sperm, and you have n for your eggs. And if you have n plus n, what do you get? 2n. So we have to make haploid gametes. Otherwise, you'd end up doubling the number of chromosomes in each generation. We're going to talk about how this happens in the next lecture. We're going to inherit our eukaryotic. Remember, eukaryote versus prokaryote. Eukaryotes are going to inherit their chromosomes in sets. Did we talk about the shape of a prokaryotic chromosome in here? I don't recall. Prokaryotic chromosomes are round, and typically they have 1. Small genome. Eukaryotes, though, we got lots and lots and lots of chromosomes, and they're linear. They're straight lines. They're going to be inherited in an entire set. You're going to get, for a human, you're going to get all 46 of them eventually. They're also going to be inherited in pairs. These pairs are called homologous pairs. They're called homologs. What does homo mean? Same. They're going to encode for the same genes. So what is at this black band on chromosome 1 is at that black band on also chromosome 1. They may be slightly different. They may, this one may make, I don't know, produces melanin in the skin and no melanin in the skin. Dark color, light color. And then you put them together and you get a medium-colored person. Does that make sense? Not the way how all genes work, but that one might work that way. So these homologs are two chromosomes that contain the same genetic coding regions and are very, very, very similar to each other. This is a homolog. This is a homologous pair. This is a homologous pair. For you, you're going to get one of these chromosomes from your dad. From your dad, the other one from your mom. So you'll have a maternal chromosome from your mom, a paternal one from your dad. They're going to encode for about the same thing because your mom and dad are not very different. They both have the same basic metabolism. They still digest things about the same way. These two chromosomes have evolved from the same chromosome. That's why they're not very different. And they match up to each other. This is where we're going to start talking about what a gene is. Now, please, if I mention things that you don't know, stop me. Because I speak science fluently. Does that make sense? And sometimes when I start talking, I slip into it. And I start saying words that I might use in another class because I teach genetics literally right before this class. And I know that I've taught them all this stuff before, so I'll start slipping into the vernacular, the words. If I say things you don't know, stop me and I will re-explain what they are. I literally would have forgotten that I didn't teach it to you yet. Does that make sense? All right, so we're going to start off at the beginning. A gene. A gene is a protein encoding region of DNA. A polypeptide encoding region of DNA. So if there's a region of a chromosome, a piece of DNA that codes for a polypeptide, that codes for a protein on one chromosome, it's going to also be on the other chromosome at about the same place, called a locus. So a gene is a polypeptide encoding region. It's found on at a of the chromosome. Questions? Your mom gives you one of your homologous pairs. One chromosome of your homologous pair. Your dad gives you the other. So you have these two haploid cells, N cells, and they come together to make a 2N diploid cell. Most homologs differ by less than 1%. That is, what you get from your mom and what you get from your dad are literally less than 1% different. The entire human species is no more than we are no more than 3% different of our entire genome than chimpanzees. 3%. That's a really small number. There's even less variation among humans. We may look at each other and say we're different, but other organisms have a really hard time telling us apart from each other. Sort of like, if you were to look at ants. You guys ever looked at ants before, crawling on the ground? They are very different from each other. But you can't tell that, right? It's hard to see. They just look like little black things scurrying around. Ants can tell the difference from one another very easily. They are less than 1% different from each other. But we have a hard time. We can tell the difference between each other. We're like, oh, look, you have this color hair, this color skin, that smell. You produce that much, I don't know, some of you guys sweat more than other people. Whatever. You've got all of these differences that we can tell about each other. One person's nose is longer than another, whatever. But those differences are very minor. Some scientists are going to study what's called cytogenetics. We're studying the chromosomes, so the strands of DNA that have a structure to them during cell division. To study cytogenetics, you need to look at what's called the karyotype. So here we have a karyotype. It tells you the number, size, and shape of the chromosomes in dividing cells. This is a human karyotype. Can you tell me, is this a boy or a girl? It's a girl. How can you tell? 2 X chromosomes. They're the same. One field of science known as cytotoxicology. What these people do is they study what happens to the chromosomes during the cell cycle when you drug things. You guys have heard of when you test drugs on animals? People are like, that's really bad, don't do that. What they're looking for are what are termed cytotoxic effects. They're looking to see if there is an effect on the chromosomes. Does it cause cancer? Are the chromosomes messed up with the introduction of this drug? So by looking at animal systems, or even non-animal systems, just cell systems, you might be able to see if there's an effect of a drug on those cells before you give it to people. So tell me, is this a boy or a girl? It's a girl. Do you notice anything about this karyotype that might make it different than other organisms? It's missing certain sets of chromosomes? They're not. They're good. So here we have 3 chromosomes. This is called trisomy 21, Down syndrome. What causes Down syndrome is having an excess of a single chromosome. Now remember, what's a chromosome made of? DNA, which means you have a lot of genes on that chromosome. Those genes are being expressed. You have too many proteins being made. All right, is this a boy or a girl? Boy, right? Why? It has an X and a Y. So anything wrong with it? Yeah, I know. Some of you are like, wait a second. It's also got 2 X's here. 2 X's and a Y. This ends up producing a person who is phenotypically, that means they look male, but they have underdeveloped genitalia, and they produce secondary female characteristics, breasts. Usually short stature and mental retardation as well, because they're producing these extra chromosomes. I'm sorry, these extra genes. Here, anything going on? Oh, sorry. The numbers on the bottom. So we've got 18, 19, 20, 21, 22, X and Y. Boy or girl? You got this. Boy or girl? What? It's a boy. Why? X and a Y. All right. Anything wrong with this organism? These aren't paired? They're paired together. These are your sex chromosomes. All the rest, though, have a pair. One of the 3 have what's called a reciprocal translocation. You don't need to know that yet. But they've swapped pieces. Now, is that going to lead to any problem? And it turns out it doesn't. The reason it doesn't is because you still have the genetic material there. You still have a total amount of yellow that's the same and a total amount of gray that's the same. But there is one thing on here that will make a big difference. 9. Why? That's right. It's missing a section. That section will lead to what's called deletion. There's missing genes. You don't have enough proteins being made. Here's a real-life karyotype. This is taken from a pregnancy that was about 3 months along. Now, I want you to know, so this thing upstairs says Mar. That means March. So don't worry about that. But do you see anything? Is this a boy or a girl? It's a boy. Do you see anything wrong with the karyotype? Number 2, you're missing a section at the top. What else? 17, another section missing at the top. 13, missing a section at the top. 6, missing an entire chromosome. They're missing a lot of genes they need to survive. This pregnancy did not go to term. Usually these pregnancies, the offspring dies very, very, very early on. You wouldn't even know you were pregnant. But the body actually is able to, or if the offspring can't produce the genes it needs, it won't be able to grow, it won't be able to develop, and it's self-aborted. It goes away. Yeah. What can it do? It depends on what part you're missing. If you're only missing a few genes, maybe you're not able to break down have some sort of metabolism problem. You might not be able to break down lactose or something. If you're missing a larger section, it could cause serious developmental defects. You might end up with mental retardation. You might end up with not being able to grow, webbing in your neck, not having the skin between, so having webbed hands, things like that. Lots and lots of any gene that's involved on that particular chromosome involved in development that's not there means that you skip a stage of development. So you could lead to a lot of problems. Yes?
So say the baby was developing normally, would it just have instantly all of those chromosomes at, let's say, conception? How come it doesn't just grow into?
So at conception, you've got the egg and the sperm. They come together. Typically, what will end up happening is these problems happen at the egg, the gametogenesis, the egg and the sperm stage, when those are being produced. Most of your eggs and sperm, if you're a young person, which you guys are, you're going to produce phenotypically normal eggs and sperm. As you get older, usually your late 30s, what ends up happening are what are termed non-disjunctions. That is, the chromosomes don't get separated into the eggs and sperm as well, which means that you're more likely to have problems with one egg will be missing a chromosome in it, or a sperm will have too many chromosomes in it, leading to these problems. That's why they call them geriatric pregnancies when you get into your late 30s, because you can lead to all of these problems very early on, and why they suggest you get tests for things. As you're younger, these aren't issues as much. Typically, when you're usually between about 17 and 24, these aren't problems. Does that make sense? So it all happens at the gametogenesis stage. All right, so we've got to get all of our chromosomes together. We need to accurately replicate our cells. To do that, the cell needs to go through a cycle called the cell cycle. It's a 4-step cycle of growth, replication, and division. We get bigger, we check ourselves out, we divide, and then we divide. This cell cycle is divided into 4 phases: G1, S, G2, and M. G1 is the first gap phase, or growth. S phase is the synthesis phase, where you copy the DNA. G2 is the second gap, where you're checking yourself out, making sure everything's functional. And M phase is the mitotic phase and cytokinetic phase. Mitosis and cytokinesis. G1, S, and G2, so growth, synthesis, checking yourself out, are collectively known as interphase. These are the phases in between mitotic phases, in between when the cell actually divides its nucleus and splits. So during interphase, the cell grows and copies its DNA. G1 is characterized by cell growth. So G1 is all about eat and get bigger. Make proteins, get bigger, do your thing. The cell is able to move from G1 phase into S phase, where it copies the chromosomes. Now check this out. In this first cell, how many chromosomes are there? Here and here. No. 1, 2. Okay, we're good on that? 2 chromosomes. You can tell there are 2 chromosomes because you can count the centers. 1 centromere, 2 centromeres. Coming over here. Not trying to trick you. Same process. How many chromosomes are here? 2. 1, 2. 2 chromosomes there, 2 chromosomes here. So beginning of S phase, end of S phase. The difference, though, is there's twice as much genetic material here. So you have 2 chromosomes in both, but twice as much genetic material at the end of S phase. If you were to make these chromosomes glow in the dark, this first phase would be half as bright as the second phase. So we've replicated our DNA. Oh, yes?
So in that step, would that be where the N goes into the
We're not worrying about that yet. Yeah, that is going to be next lecture. After S phase, the chromosomes are still joined together. Vocabulary term is going to come up in lab. The center part's the centromere. That's where they're connected. Each leg, or not leg, but each copy. Here's one copy. Here's an exact copy. Or joined together at the centromere. Each of these are called sister chromatids. They encode for the exact same genes in the exact same way. They are identical copies. This side is the exact same as that side, as long as everything worked out right. So you have two sister chromatids joined together by a centromere, and that makes this chromosome. So again, by the end of S phase, you've got twice as much genetic material as you started with. Two sister chromatids joined together by a centromere. That lets you move into G1 phase. Right after you get through S phase, the cell has to produce all the proteins that it needs for sorting and cell division. You're getting ready to reproduce. So you have G1, which is cell growth, S, which is synthesis. You create more, you double the amount of genetic material, and then G3, which is where you're checking yourself out. There's another phase called G0. It's not called go. It's called G0. This is an alternate phase. This is where cells go when they don't work out so well. This is where cells go when there's damage. You have too many chromosomes present, too few chromosomes. You didn't copy things right. This is like timeout for cells. You're like, no, look, you got to calm down, go sit in a corner, fix yourself. If a cell enters into G0, it can work on fixing itself so it can get back into the phase. So this is sort of an alternative phase that cells go into if they fail. So G1, S, and G2 are all interphase, connected together to get from growth to copying the DNA to checking the DNA out and producing all the proteins you need. If you do not get through those phases satisfactorily, if you're not big enough, if you're not, if your DNA is broken, if you don't have enough proteins to proceed, you enter into G0 phase. From interphase, you start leading into the mitotic phase, or M phase. This is probably what you've heard a lot about. There are two distinct parts of M phase, mitosis and cytokinesis. Mitosis is where you sort the cell's nucleus into two daughter cells. Into two daughter nuclei. Now, this is the big difference between what a lot of people learned in high school or in middle school and what they have to learn now. Mitosis is not cell division. Mitosis is the division of the nucleus. Cytokinesis is the division of the rest of the cell. But people tend to use those terms interchangeably, and they are really two distinct things. I'm going to go through mitosis, the division of the nucleus. At the same time, while mitosis is happening, you have cytokinesis, which is the division of the rest of the cell. During mitosis, you're going to take the sister chromatids that you created during S phase and split them in half, so that each of the daughter nuclei has the same number of chromosomes as the parent nuclei. So that's sort of the big picture of mitosis. Cytokinesis is the division of the cytoplasm and the organelles. That's where you sort of split your Golgi apparatus. You split your endoplasmic reticulum. Some mitochondria, some chloroplast end up in one daughter cell, some end up in the other. But you're dividing up the cell. For a typical 24-hour dividing cell, you have G1 phase, takes about half its life, 11 hours. S phase would take about 8 hours, a third of its life. G2 phase, where you're checking yourself out, takes about 4 hours. and M phase takes about 1 hour. Now, not all cells work on 24-hour cycles. Some work on 8 hours, some work on 1 hour, things like that. But this just gives you a basic idea. Most of the cell's life, the vast majority of it, is in interphase. Mitosis is only a very small amount of it. It's like with you guys. Most of your life is not actively reproducing. Most of your life is probably eating. I mean, probably most of you right now are thinking, not thinking, I'm going to go and reproduce. No, most of you are thinking, I'm really hungry. Or I'm really tired. Or I need to get more resources. Reproduction is just a small amount of your life. This cycle is regulated through these, and this is coincidentally enough, through proteins called cyclins. The cell cycle regulated by cyclins. And you can see here that cyclin D1 is abundant during G1 phase. You're producing a lot of it. As cyclin D1 drops in amount, cyclin E increases. When you get cyclin E, a lot of cyclin E, cyclin A turns on. And as cyclin A turns on, it turns off cyclin E. As cyclin E peaks, cyclin B turns on, and cyclin A begins to drop off. Here's a graph. So you can see cyclin D rising, and as cyclin D peaks, cyclin E kicks on and begins to peak. And as cyclin E peaks, cyclin A turns on. As cyclin A peaks, cyclin B turns on. As cyclin B peaks, cyclin D decreases. And you end up with a cycle. And here's the cool part about graphs. You looked at this graph and you said, yep, do you guys all see the trend? Do you understand the idea of looking at the concentration increasing with cyclin D over this is time? You understand how to read a graph, right? If you can understand how to read a graph, you don't even realize that the graph is written in German. Does that make sense? A good graph can overcome language barriers. And they didn't just misspell concentration. In order for the cycle to work, you need to constantly check yourself out because you never want to reproduce a broken cell. That leads to cancer. You have what are termed checkpoints. 3 points in the cell's life where it looks at itself and says, am I good enough? And you know you've all been through that phase. You looked at yourself in the mirror and you critically evaluated yourself and you thought, am I really going to do this? Am I good enough for this? It's that Stuart Smalley thing. Am I good enough? I'm smart enough? During G1, the cell looks at itself and says, is it safe to divide? Do I have enough resources? Am I big enough? If it's not, it enters into G0. If the DNA is damaged, it enters into G0. During G2, you've just synthesized your DNA. You've just copied your DNA. A whole bunch of these proteins run along the DNA. And they make sure that each of the sister chromatids, each of the sister chromatids attached to each other are absolutely identical. If the DNA is damaged, it enters into G0. During the mitotic phase, M phase, you have this point called metaphase, where where all the chromosomes line up along the center. And if the chromosomes aren't ready, if they're not lined up properly, the cell enters into G0. G0 is there to stop broken cells, to stop cells with damaged DNA from reproducing themselves. If you don't have a checkpoint, if a checkpoint fails, if your checkpoints are not working, that means that broken cells can begin to divide and divide and divide over and over again. And when these broken cells divide, you end up with these huge tumors. Cells that are undifferentiated but don't know when to stop. They keep producing more broken cells. This was a tumor that was pulled out of a woman. She didn't even realize she had it. She was a very large woman, in case you couldn't tell. But she had this. This was a tumor that was sort of up. That was at the top of her thigh. About 20-pound tumor was removed from her. That is the result of cells not being able to go into G0 phase. One cell screws up. Just one. If one cell screws up and is constantly loses a checkpoint, then its offspring the two cells will match that first cell, and they will always screw up, and their offspring, and so on and so on and so on. Exponentially, this mass grows. Ultimately, cancer is a problem of the genes. Cancer is a problem of being able to faithfully replicate your DNA. It's just a ticking time bomb. Truthfully, over time, this could happen to anybody. Obviously, there are certain environmental conditions that can increase the risk for cells not being able to enter into G0 phase. Not being able to enter into G0 phase. Smoking, tobacco, carcinogens, all sorts of things can increase the chances of cells mutating and skipping checkpoints. Once they do, it's all downhill. So we need to accurately and faithfully reproduce our DNA. And when we do that, our cells are able to divide. We use mitosis, the division of daughter cells, and cytokinesis for cell division. And cell division is useful for asexual reproduction and in certain prokaryotes, but it's also good for you for repairing wounds, for repairing your body. So mitotic cell division creates two cells that are identical to the parent. This leads to asexual reproduction, where an organism makes an exact copy of itself. This is an amoeba dividing. It's big enough it can divide. As the chromosomes begin to divide, you're going to have those two identical sister chromatids. So you've got your centromere and your two sister chromatids. Surrounding the centromere of a chromosome, you have a structure called a kinetochore. Here you go. Chromosome has kinetochore. Kinetochore is a protein belt. These microtubules, you guys remember what microtubules are? What are they? Right, motor proteins walk along them. They're rails, right? They help things move around the cell. Microtubules will attach to this protein belt, this kinetochore, around the centromere, allowing them to pull or push the chromosomes wherever they want in the cell. This is what moves the chromosomes around. So you have the mitotic spindle forming at the centromeres. They come down, they bind to the centromere. I'm sorry, did I say centrioles here or centromeres? All right, sorry, century, right? Microtubules start at the centrioles, or the microtubule organizing region. They're then going to attach to the kinetochore of the chromosomes that surrounds the centromere, letting them push or pull chromosomes into place. Sort of like if you want to drag somebody around, if you grab them by their belt, they have to go where you pull them, because that's their center of mass. So we have to get through mitosis. All of this is building up to the phases of mitosis. Now these are phases I'm sure you've all memorized at one point in your life or another. It's the idea of interphase, which is all G1, S, G2. Prophase, metaphase, anaphase, telophase. Was it PMAT is what people say. The first step of the cell cycle, of course, is interphase. The genetic material is replicated, the cell grows, the chromosomes decondense. They're open weaves, they're just chromosomes. Then you start moving into M phase, the very short part of the cell's life cycle. It starts off with prophase. Pro means first. The sister chromatids condense. So here you've got your sister chromatids that you can see. They've condensed so you can see the chromosomes. And the mitotic spindle begins to form at the centrioles, or the centrosomes. The nuclear envelope begins to dissolve. It starts to break up into vesicles. Then you enter what's known as prometaphase. During prometaphase, the whole point of this phase is for the microtubules to attach to the chromosomes. So microtubules attach to the kinetochore around the chromosomes. It's prophase, followed by prometaphase. Next up, we have metaphase. During metaphase, the microtubules push or pull the chromosomes into position, getting them to line up along what's called the metaphase plate. So here they're a straight line across the center. From there, you enter into anaphase. During anaphase, the chromosomes, the sister chromatids, are pulled apart into individual chromosomes. They get pulled to either end of the cell. And then during telophase, the chromosomes begin to decondense, the nuclear envelope reforms, and you have two distinct nuclei in the cell. Each of these are an exact copy of the other. That's really it for mitosis. I mean, prophase, prometaphase, metaphase, anaphase, telophase. Will you need to know those? Yes. Are those going to be an essay question? Yes. At the same time mitosis is occurring, you have what's called cytokinesis. Cytokinesis is where you're actually dividing the cell. You're actually dividing the rest of the material. Mitosis is the division of the nuclear material. Cytokinesis is the division of the rest of the cell. The first thing in animal cells that happens is a ring of actin filament. So remember, actin causes actions, forms around the cell, the whole cell. So it's like a belt around the cell. And then that actin gets tighter and tighter and tighter. As it gets tighter, the cell squeezes into two individual daughter cells. With any luck, what should have happened at this point is, over time, a nucleus forms over here and a nucleus forms over there. You get some Golgi over here, you get some Golgi over there. Does that make sense? You're just splitting that material. That works really well in animal cells, but it doesn't work well in plant cells. Why would this not work in plant cells? What do plant cells have that animal cells don't? A cell wall. Actin filament isn't strong enough to break through a cell wall. So instead, with plant cells, what you form is what's called a cell plate. So the cell plate forms between two daughter cells, between the two nuclei. What ends up happening is the Golgi apparatus starts shipping a whole bunch of cell wall proteins to the middle of the cell and just dropping them off there. Have you guys ever built a dam in a creek before? Or tried to dam something up? When you do that, you start off with one rock. You take the rock, you put it in the water. Does it do anything? No. Water still flows around. It takes you dropping off rock after rock after rock in the center. The Golgi is dropping off vesicle after vesicle after vesicle. And over time, it builds up until you have a wall blocking off the creek. In the cell, it builds up until you've created a complete cell wall between these two cells. So in animals, you're going to get that cleavage furrow forming, but in plant cells, you're going to get the cell plate forming. It's absolutely vital for multicellular organisms like yourself to produce identical chromosomes, to produce identical daughter cells. If you don't, each cell in your body would have a different set of directions. You'd be telling it to do different things. Does that make sense? Your brain cells would be different than your heart cells, would be different than your stomach cells. And you don't have a cohesive set of directions. It'd be like trying to cut and paste together directions from many different sets of electronics. You wouldn't be able to do it. Sure, there's some similarities, but ultimately the differences would result in the whole system failing. So for you, you need to produce cell after cell after cell that's the same. When you're repairing your cells through mitosis, you get a cut. You need to heal the wound. The wound heals because cells rapidly divide on those edges until it closes back up. I mean, it may not work like as fast as it does in Wolverine or something like that, but it still totally works. Eventually, you heal. And I have armadillos. I don't know why I put armadillos on this slide. There was seriously a reason. It had to deal with my... Oh, that's what it was. Okay, so you guys know what an armadillo is? It's sort of like an armored rat. Okay, armadillos are a menace in Texas and Florida. They're all over the place. Anybody ever seen an armadillo? Okay, if you've ever been in southern states, they're everywhere. Typically, you find them on the side of the road dead. I just want to let you guys know, armadillos always produce quadruplets. 4 absolutely identical offspring. Every single time. The egg and the sperm fuse, they split to produce twins, and they split again to produce quadruplets. Identical. These 4 armadillos have the exact same genes. They are literally clones of each other. So, if you were to ever run one over, don't feel bad about it, because there are 3 more just like it. There you go. That's the takeaway. Any questions? All right, please don't forget these content review questions. They will help if you go through and write out the answers. I'm not going to collect them, but people are always asking, what can I do better? I don't like my grade. Please keep up with these content review questions. Just write out the answers if you will, and then study those for the exam, and it will help a lot.